Hi, everyone. Welcome to and thank you for coming to another Fragile Nucleosome seminar series. This is a bit of a special edition of Fragile Nucleosome. This is our last set of presentations for our spring seminar series. We'll be taking the summer off and then kicking everything back into gear in September. We will be sending out in the next week or so a new edition of our sign-up sheet. So if you know anybody or if you yourself would like to volunteer or nominate someone to present in the seminar series, keep an eye out for that. And let's see. Also, we had three amazing speakers that we wanted to get in before we took the summer off. And so today is going to be a special session where we'll go about 90 minutes. Each of them will be given a full 20 minutes for their talk. And I'm really excited to be the person to introduce Raquel Fueyo, sorry. She'll be giving the first talk today. She originally, I guess she started off her scientific career in Spain, in Barcelona, doing her PhD with Marian Martinez Balbas, and briefly with Alvaro Rada Iglesias, where she did some really cool work looking at JMJD3 and how it functions in regulating transcription and enhancers. Then about 5 years ago, she moved to California, to Stanford, to do a postdoc with Joanna Wysocka. And there she is now a leading edge fellow. And excitingly, she said that we're allowed to say that in a few short months, or I guess at the end of the year, she'll be starting her own lab at the Max Planck here in Europe. So that's very exciting. And so I think today we will get to see a little bit of the work that she's trying to get published now and stuff that will become the future Fueyo lab. So Raquel, please take it away.
Thank you. Thank you for this very kind introduction and this very nice initiative that is Fragile Nucleosome. So I will start my talk by retro-jumping 50 years into the past. So the 50s and 60s were very prolific on virology discoveries, and it was at that time that analysis of healthy tissues identified viral particles that were not the product of acute infections, but they were product of vertical transmission. So this work led to the observation, for example, of viral particles in human placentas, hinting that viruses may have something to do with human embryogenesis. The human genome contains 50% of transposable elements, and transposable elements are these pieces of nucleic acids that at some point in evolution, or even now, had the capacity of mobilizing from one genomic region to another. This percentage contrasts with the 2% of protein-coding genes. Protein-coding genes are highly conserved between species, however, transposable elements are not, suggesting that they could be contributing to a species-specific functions. So within transposable elements, I'm particularly interested in endogenous retroviruses, and these form around 9% of our genome. So I may refer to them as ERVs, so just so you know this acronym. So ERVs originated from exogenous retroviral infections that occur in the germline, and they could be propagated from parents to offspring in a Mendelian fashion and potentially getting fixed in the population. At the time of insertion, the open reading frames for the viral proteins are flanked by 2 long terminal repeats. However, during evolutionary time, many mutations can accumulate over these open reading frames, limiting the capacity for these viral proteins to be properly translated. Another phenomenon that occurs is that the long terminal repeats can recombine through homologous recombination and give rise to these solo LTRs. So I want to remark that in humans, this is the main shape of endogenous retroviruses, as solo LTRs. ERVs have affected human evolution and physiology by providing a unique repertoire of coding and non-coding sequences to the genome. So the best known case is the co-option of this fusogenic protein from the ERVW that works as a surface receptor and allowed placentation. However, ERVs can also contribute to disease, so genetics, the environment, age can all affect cellular homeostasis, and this regulation of this homeostasis can lead to the upregulation of ERVs that has been found in, for example, oncogenesis, neurodegeneration, or autoimmune disorders. In humans, ERVs no longer retrotranspose, but their long terminal repeats that were the original promoters and enhancers for the viral genome can still work as enhancers and promoters in our own genomes. So this is very common, for example, in stem cells and the germline, and this is called co-option, but we still don't know whether this co-option has a function or what is the extent of this functional impact. So if we want to answer this question, I think the preimplantation stages of embryogenesis is one of the best models for it, because ERVs are epigenetically silenced in most somatic tissues. However, they become active during preimplantation, for example, in humans and in other mammals. So I want to remark that when I'm talking about preimplantation, I'm talking about this series of events that happen from the moment of fertilization to approximately 6 days or 7 days when the embryo will attach to the uterus. So at this particular stage, the blastocyst stage, we have 3 different lineages present: the epiblast that will give rise to the embryo proper, the hypoblast that will develop into the yolk sac, and the trophectoderm that will give rise to the placenta. So now coming back to endogenous retroviruses, one key aspect is that the pervasive DNA hypomethylation that occurs during preimplantation in mammals leads to the upregulation of different families in a lineage-specific manner. So this suggests that endogenous retroviruses could be playing a role in embryogenesis. And so to understand the impact of ERV's co-option in embryogenesis, I decided to work with a long-known endogenous retrovirus in the Vysotsky lab that is called HERV-K. So this endogenous retrovirus is one of the youngest in the human genome. It started infected after the split of wild monkeys from apes, and then it continued infecting after the split of humans and chimps. And in fact, there are some insertions that are specific to humans and some even polymorphic in the population. So HERV-K can come in 2 different flavors as long terminal repeats that are called LTR5HS. So I may refer very often to LTR5HS. So this is just the name of the long terminal repeat. Or we have around 100 proviruses that can still encode viral proteins. So as other ERVs, endogenous retrovirus HERV-K becomes active at the 8-cell stage, and then it peaks in the blastocyst. So the expression is localized to the epiblast, to the hypoblast, but is not present in the trophectoderm. So not only transcripts, but also viral proteins can be detected in human blastocyst cells, and even viral-like particles were found in vesicles of human blastocysts, and this previous work from Joanna's lab that I found very fascinating. So once again, this suggests that our own human embryogenesis is occurring in the presence of viral products. So I think all of this shows that HERV-K is active in human embryogenesis, but does it have any function? So if we want to answer this question, it would be ideal to do experiments using human embryos, but obviously experiments with human embryos entails practical and ethical and legal challenges. So having enough numbers to perform functional assays is very difficult. However, my postdoc was perfectly aligned. I was very lucky with the development of protocols to generate this type of organoid that is named blastoids and that mimic the human blastocyst. So they recapitulate the natural blastocyst. We can get hundreds and thousands in the plate. I optimized the functional assays, epigenetic and genetic manipulation, and you can even try to mimic the implantation of these blastoids into plastic or endometrial organoids. So they really open a lot of opportunities. So to generate these structures, you need to use human naive pluripotent stem cells. So these are not your typical embryonic stem cells. Those can only give rise to embryo tissues. Naive pluripotent stem cells can also derive into extra embryonic tissues, like, for example, the trophectoderm or the yolk sac. So the protocol starts, you put certain number of human naive pluripotent stem cells in the presence of cytokines, and after 5 or 6 days, you get these structures as I'm showing here in this video. So I decided to use this system to address the function of HERV-K, and for that I needed to optimize genetic and epigenetic manipulation in naive pluripotent stem cells and human blastoids. So the system that I implemented to test HERV-K function started from human naive pluripotent stem cells, where we integrated dCas9 fused to the transcriptional repressor CRAB. And then we integrated a set of guide RNAs that don't target anywhere in the genome as a control, or a set of guide RNAs that can target LTR5HS elements. So you need to think that because these elements are product of retrotransposition, the sequence is very similar. So with a bunch of guide RNAs, we can target around 90% of the insertions. So the idea is that when we induce the system, CRAB will bind and will deposit H3K9 trimethyl and will repress the LTR5HS elements. So to test if this system is working, we perform cut and run in non-targeting and LTR5HS targeting condition. And this is what I'm showing here. So you have this browser capture where you can see that in the LTR5HS repression condition, the elements gain H3K9 trimethyl, and more systematically, I'm showing in these heat maps where each row represents one insertion of the LTR5HS element, and you see that when we induce the system, they gain H3K9 trimethyl, and also we see a decrease in the transcript originating for LTR5HS. So this system works very well for repressing. So then to test if the pluripotency of the developmental potential of these cells was affected, we moved to a human blastoid system, and using control and LTR5HS repressed clonal cell lines, we trigger blastoid formation. So first we analyzed the blastoid formation potential at different levels of HERV-K repression. So first with the control clones that I'm showing here on the left, you can see that the blastoids can form without problems. However, we observed that when we repress LTR5HS elements, the blastoids can't form, they do not cavitate. So this suggests that HERV-K activity could be impacting human development. So now to show it more systematically, I'm plotting here quantification of the blastoid formation potential of around 24 clonal cell lines control and 24 where we are repressing LTR5HS elements. So in the x-axis, you're seeing the levels of LTR5HS activity, and the y-axis represents the blastoid formation potential. So I hope you can appreciate multiple things. So first, that the LTR5HS levels correlate with the blastoid formation potential. And second, as I showed in the video, when we fully repress LTR5HS, the blastoids can't form at all. So as a control, we show that at high levels of LTR5HS activity, neither the control nor the LTR5HS repressed clones can form blastoids, have any problem forming blastoids. So this shows that the effect is specific and that this is not due to experimental variability that is very common because these models are very new. So some of you may have noticed too that at medium levels of LTR5HS repression, the blastoids can form, but we ask whether these blastoids are normal and whether we could use them to investigate the impact of HERV-K on lineage determination. So to deepen into the potential mechanism of medium repression blastoids, we perform a PIP-seq, that is an alternative to a single cell with 10x. We dissociated the cells from control blastoids and blastoids where LTR5HS is repressed, and then we analyzed the transcriptomic profiles. Because blastoids are in vitro structures, to assign the different lineages, we need a reference data set from human embryos. So for that, we use a collection of data that has been published from the field. So this starts from the zygote to pre-implantation and post-implantation stages. And first, we projected the control blastoid cells, and we confirmed that they have all the analogs to the human blastocyst. So they contain epiblast analogs that in vivo will give rise to the embryo proper, analogs to the hypoblast that will develop into the yolk sac, and analogs to the trophectoderm that will develop into the placenta. But now I guess the question is, what happens when we repress LTR5HS elements? Do we see any defects? So the answer is yes, and these results have been confirmed by immunofluorescence, but I don't have to show it today. So we detected two main defects. So first, we observed that when we repress LTR5HS elements, the blastoid's epiblast gets stuck in an earlier state that is actually defective. It's not any real state when you take the transcription factors. And second, we detected that is a complete hypoblast loss, and this makes a lot of sense because the hypoblast derives from the epiblast. So if the epiblast is already defective, so I guess you are losing those cells. So these results showed that in those cells that HERV-K is expressed, the epiblast and the hypoblast, we observed defects, and that HERV-K is affecting lineage determination. But what is the mechanism of this? So to answer this question, we started performing differential gene expression analysis, comparing the epiblasts of non-targeting control blastoids and those of where LTR5HS is being repressed. And this analysis detected a lot of dysregulated genes, mostly downregulated genes. But if you go to test the gene ontology categories of these genes, they don't have anything in common. You won't find any. However, they have one thing in common, and is that most of them lie within 250 kilobases of an LTR5HS insertion. So this is very close. You need to think that usually an LTR5HS element is around 1.62 megabases far from a gene. So these genes are close to LTR5HS element, and this is consistent with LTRs working as enhancers for these genes, and this has already been shown for LTRs and other type of endogenous retroviruses. So to verify that these elements are actually working as enhancers, we perform a series of genetic lesions of LTRs followed by RT-qPCR of the putative regulated genes, and we show that indeed most of them are working as enhancers. However, one such deletion, the copy that is near the SYNfinger 729 gene, shifted the way that we now think about endogenous retroviruses, and now I will dedicate the rest of my talk to it. So deletion of this single LTR5HS copy, the one that is near the SYNfinger 729 gene, leads to the repression of this gene. However, we also saw a striking defect on cell proliferation. So in other words, the cells started growing very slow, but we have only deleted 1 kb that is an LTR5HS element. So as you know, as a lab that we know about enhancer biology, this was very surprising for us. So I became very puzzled by this insertion, and I decided to check the genomic landscape. So first I realized that this LTR5HS is only present in the genome of humans. And the gene itself is very new too, so it appeared around primates. It's not very, very clear. So far we have only detected SYNfinger 729 expression in naive pluripotency in humans and not even in chimps. So, but even though here I think I should remark that there are no naive pluripotent stem cells from other species that are not human, chimpanzee, or macaque. So it could be that it's expressed in orangutan, it's just that we don't know. But so far we have only detected expression in humans. And this made me think whether a single LTR5HS insertion may be necessary for human embryogenesis. So we took wild-type cells and cells where we have deleted this LTR5HS element, and we triggered blastoid formation. So with multiple clones, I observed that in the absence of this single LTR5HS, the blastoids can't form. But to rule out off-target effects, we performed rescue experiments by introducing a transgene, and we were able to partially rescue this phenotype of blastoid formation. So this shows that a single human-specific insertion is essential for blastoid formation. So this result made us wonder about the function of this SYNfinger 729. So most of you probably know that SYNfingers belong to this family of transcription factors, and that currently the most accepted idea is that they evolved to repress transposable elements. So they have this ability of recruiting CAP1, also known as TRIM28, and they also recruit the pregenetic repressors and lead to the repression of transposable elements. So what is the SYNfinger doing in naive pluripotency? So to answer this question, I engineer human naive pluripotent stem cell lines, adding a degron tag and 2 HA tags to be able to perform immunoprecipitations. And in this system works very, very well. So when we add the D tag, we can degrade the protein as early as half an hour. So because the zinc finger 729 is a transcription factor, we perform ChIP-seq to analyze the genomic locations. And this experiment was super surprising because we see that it binds to at least 46,000 regions in the genome. And surprisingly, most of them are not transposons, but are actually promoters. Almost 50% of them are promoters. So these peaks are specific, as you can see here, because when we treat with VTAC and we degrade the protein, they disappear. So, of course, at the beginning, we expected zinc finger to bind transposable elements, but why is the zinc finger binding to more promoters? So we performed motif discovery analysis with many softwares, and we found many different results, but all of them have one thing in common, and is this very high affinity for GC-rich regions. In fact, as I'm showing at the bottom plot, the promoters that are bound by zinc finger 729 display higher GC content. So at the gene expression level, so what is happening? So when we acute deplete this protein, we don't see any changes on transposable elements, so only in genes. We see at short term, when we deplete zinc finger 729, the changes that are consistent with an activator rather than a repressor function, something that is very interesting and that now we are investigating. And then after 24 hours, we see both genes that are upregulated and downregulated. And so this suggests, because these are almost 2,000 genes, that zinc finger 729 is a key transcription factor in naive pluripotency that has not been known before. So then in this last volcano plot, I'm showing that almost 40% of the genes are regulating directly at the promoter level. And I was very stringent with the promoter levels. We are talking about 1 kb around the TSS. And we observed that most of the genes that get downregulated, not most, but many of the genes are related to cell cycle. So this is consistent with the phenotype that we detected. And also we see many other genes related to metabolism or other cellular basic functions. So mechanistically, we know both activation and repression are operating through TRIN28, but we don't have time to show the data today. And yeah, because I'm running out of time, I'm gonna go and talk about this model. So I hope I've convinced you that viral elements have to do with human embryogenesis. And today, as a summary of the model, I've shown you that a human-specific endogenous retrovirus named LTR5HS activates the expression of a very young transcription factor, the zinc finger 729. This zinc finger is the zinc finger with the highest number of domains in the human genome, and it has affinity for GC-rich regions, binds to promoter of cell cycle and basic cellular functions, and those are known to be very GC-rich. So this mechanism seems to be essential for naive pluripotency and blastoid formation, and who knows, maybe for human embryogenesis too. So these results show the power of using human blastoids to reveal human-specific features of development, and they also show how transposable elements have remodeled gene regulatory networks, and they endow all cellular environments with new functions. So very quickly, I want to thank the people in the lab, especially Joanna, that is a fantastic mentor, my sources of funding. And this very last minute, as Jackson said at the beginning, I will be starting my lab in the Max Planck Institute of Molecular Genetics, where we will use these human embryo models to study the impact of endogenous retroviruses and embryogenesis. So just to say that I'm hiring at all levels. So if you're interested in this work and you're interested in coming to Europe and Berlin, that is a very funny city, just let me know. Thank you.
Awesome. Thanks so much, Raquel. That was great. To the audience, if you have any questions, feel free to put them in the Q&A, or if you'd like, you can raise your hand and I'll unmute you. We have time for at least a few questions, and while we wait for them to come in, I'll start. Raquel, do you think is CNF 729, how does its expression relate to zygotic genome activation? And is that something you can even model in the blastoids?
Right, so it starts being expressed around the morula state, and that's exactly the moment where HERFK becomes active. So we have detected a few transcripts, but we think they are maternally deposited. So the expression really correlates with endogenous retrovirus. It's not there before.
And so then do you think this is like in thinking about like the evolution of this or how it could be co-opted for human evolution, do you think it's given some advantage to
Yeah,
humans over
yeah,
lower primates or other animals?
yeah. Yeah, this is a very interesting question and very difficult to answer. So first of all, we started this project thinking that these elements will not be essential. So this was a big surprise. But if you think about it, these endogenous retroviruses, to be very successful in embryogenesis, they needed to co-opt a lot of the pluripotency factors. So this made them very, very strong enhancers. So what we believe it happened is that the insertion of the element suppose such a strong enhancer that whatever ancestral mechanism was there may have disappeared, and then it activates the expression of this zinc finger during this element. Then the zinc finger is very special because it has 37 domains as compared to 13 on average. So we think this element started being expressed, this gene started being expressed, and it became very good at binding promoters, probably substituting another ancestral function. So who knows what is doing this in mouse, for example.
Right. And then, something in the Q&A. Okay, got a couple questions in the Q&A. We have time, I think, for two more. So Antoine asks, or says, amazing talk. What do you think would happen if you overexpress 729 in other species?
Yeah, so now that I'm done with this job market thing, so I want to do those experiments in mouse. And yeah, so I don't have the results. So my bet would be, because we see that it binds to promoters in humans and also it brings TRIM28. So my bet would be that probably will bind to some promoters in mouse. The question is, will it recruit TRIM28 or not? Or would it be able to displace whatever mechanism is doing this in mouse? So yeah, this is a very interesting question and we are working on that, yeah.
Well, actually, I think we need to move on to the next talk. If anyone has further questions for Raquel, please put them in the Q&A. Since we have three speakers today, please, if you ask any more questions, lead them off with the speaker's name so we know who they're being pointed to. But once again, thank you very much, Raquel, and I will now pass it over to Skye to introduce our next speaker.